Hey everybody, we are looking at section 13.2, which is going to be all about a little bit more of the history involving DNA, but also now the structure of DNA. So the parts that make it up, the elements that we see, the monomers, the polymers, the types of bonds, all those things that we haven't talked about really for a little while. We're going to come back and look at a lot of that content again here in section 13.2. So not a long section, but there is quite a bit of really important information in this. So go ahead, make sure you've got your heading, which is again section 13.2, structure of DNA, and then we'll go on. So for the objectives, I've got 3 here in this section. So we're going to look at the chemical components of DNA, so meaning again the elements that we find in this molecule. We're going to look at how scientists were able to discover and kind of talk about and communicate the structure of DNA. And then we're going to look at what we understand about DNA given the models that have been created in regards to their structure. Okay. So one of the things that we mentioned and talked about back, I believe in chapter 2, when we first started looking at biomolecules or these organic molecules, was that nucleic acids are made up of monomers called nucleotides, and hopefully you remember that, and if you do, that's going to be helpful. If not, of course, you've got it right now, so you can spend time reviewing that here. So the nucleic acid itself is actually the polymer. It's made up of many nucleotide monomers linked together, and they link together, as in all of our biomolecules, with covalent bonds. All right, so DNA and RNA are examples of nucleic acids, so they are actually polymers. They're each made up of long chains of nucleotides, and again, they have those strong covalent bonds. The only real difference between DNA, and we'll come back to this, but the only real difference between DNA and RNA is that DNA has a sugar called deoxyribose, which you see right here. This little pentose sugar, it's got 5 sides. And RNA has a sugar called ribose, which looks the same in a diagram like this. It also has 5 sides. The difference is that deoxyribose is missing an oxygen here, and ribose has an oxygen there. So we say deoxy like it's missing one, okay? You should just know that these are the 3 parts of a nucleotide that we find in DNA. So we have nitrogen-containing bases, we have a deoxyribose sugar, and we have a phosphate group. All right, and you absolutely should know those 3 parts. I would even say it would be really good if you would draw those in your notes here, because if I was in class teaching this with you, I would ask you to draw them, okay? So you're going to want to have the nitrogen-containing base, the sugar, and the phosphate jotted down, okay? And phosphate is something that you should maybe feel like it's kind of familiar, because we talked about phosphates a lot when we went through cellular respiration and photosynthesis in chapters 9 and 10. There, we discussed a molecule called ATP, which was adenosine triphosphate. So phosphate is reappearing here. Phosphates were also found in phospholipids. Remember, they have a phosphate head group. It's the same molecule. It's just a phosphorus atom with 4 oxygens around it. So it's PO4, okay? You don't have to know that chemical formula, but just recognize it is the same thing we've been talking about, okay? So when we think about these nucleic acid monomers, right, these nucleotides, we know that in DNA there are 4 different types of nucleotides total. That's it. So it doesn't get more complicated than that. The 4 different types are named based on their nitrogen-containing base, or what we sometimes call their nitrogenous base. So the nitrogen-containing bases can be one of 4 different types, and those are labeled for you here. So they can be thymine, cytosine, guanine, or adenine. And so when we go to simplify those to talk about these more simply than having to say that whole word, we tend to talk about thymine as being represented by T, cytosine is a C, adenine is A, guanine is G. For these, all right, again, when we think about our nucleotides themselves, if I flip back, these 2 are always going to be constant. Those do not change between any of the 4 different types of nucleotides. The only thing that varies here is this. So again, either this nitrogen-containing base can be an A, a G, a C, or a T. All right. So what I want you to get from this on this one, you don't have to draw the structural formula. You don't even have to draw the models. But what I would like you to make sure you have is these labels up here up at the top. All right, pyrimidine and purine, and I'll describe those here in a second. And then the names of the bases that fall into each of those categories and their abbreviations, okay? So if we talk about the pyrimidines here first, these are nitrogen bases that contain just one single ring. Their whole structure is consumed by this one single six-sided ring. So you can see thymine, here are the actual elements that we find. When we simplify that and just kind of represent our carbons, we see it's basically just a single ring, right? There's some things that come off of it, but for all intents and purposes, it's just one continuous ring. So thymine is in that category. Cytosine is also in that category, and if you notice, it looks very similar. But if we compare that to our purines over here on the right, those have double rings. And this does make a difference. We're not just saying this because, I want you to know it, but you do need to know this so that you understand how these things pair up in a molecule of DNA. So adenine has 2 rings, and guanine also has 2 rings. I am going to ask that you keep these things straight and that you can tell me what are our examples of pyrimidines and what are our examples of purines. And so the way that I remember that, when I see these, thymine and cytosine have Y's in their name, and pyrimidine also has a Y in its name. So if I can keep straight that thymine and cytosine fit in this category, remember I only have 4 total, so that means my other 2, adenine and guanine, have to fit with purines, okay? Pyrimidines have a single ring, purines have a double. The way I remember that is actually to think about it in the reverse. So purines have purines have a double ring, but they're actually the smaller of the 2 words, right? Purine is a smaller word, but it actually represents the bigger structure. Pyrimidines, on the other hand, have the single ring, even though pyrimidine is the bigger name, okay? So I would find a way to keep those straight. If that helps you, then by all means use it. If it doesn't, and you come up with another better way, then certainly, let us know, because that would be helpful maybe to your peers, okay? So this is a polymer of nucleotides, right? You can see if I were to highlight, this would be one nucleotide. These are the 3 parts here. Here's another nucleotide, another one, and another one. So in total, I have 4 nucleotides here. Now, we have way more than 4 nucleotides in a base, right? We're talking about thousands or even hundreds of thousands of these things all in line in a specific order. But you can see here how they differ. A, G, C, and T, and their sizes, again, are different. So when we look at these, again, this is one single nucleotide. And then between those nucleotides, I have these nice covalent bonds, right, represented by these nice straight connected lines. I have no dashed lines here because we're not dealing with weaker interactions. These are all covalently linked together, okay? So this part of my DNA molecule is actually called the sugar phosphate backbone. So my phosphates, again, are these little blue spheres. My sugars are those pentose 5-sided deoxyribose sugars, and notice they alternate. So as we go down this DNA polymer, we like to call it a double ladder or a spiral staircase, and we'll see why here in a few minutes. But as I go down this ladder, notice how they're alternating, right? Phosphate binds with sugar, which binds with phosphate, which binds with sugar, and so on and so forth, okay? And so in a diagram of these things, you have to represent them properly so that you can see how those, how those kind of alternate going down the backbone. All right, so in talking again for a minute here about scientists and what they had to do with our discovery and understanding of this molecule, there was a guy by the name of Erwin Chargaff who helped to identify how those nucleotides we just talked about actually interact in a molecule of DNA, because we know that somehow sequences have to be maintained. Right? Somehow what parents gave you, we know you can express, but that you can also pass that down to your children. And somehow we have to be able to maintain that. So DNA itself is not just, this single-stranded molecule. DNA is actually double-stranded, more complicated molecule. So Chargaff was a chemist, and Chargaff helped to kind of quantify per sample of DNA. So he would assess DNA in lots of different living things. He would obtain those DNA samples and he would purify them. And then he would kind of calculate through chemical means the amounts of A's, T's, G's, and C's. So the amounts of each of these nitrogen-containing bases, or in other words, the amounts of each of these nucleotides. And what he noticed was that in any sample of any DNA from any living thing, the amount of A was always equal to the amount of T, and the amount of G was always equal to the amount of C. So what that told him is that wherever in a DNA molecule we have an A, there must be a T next to it or very close to it. Wherever in a DNA sample we have a C, there must be a G that kind of interacts with it also. It does not mean that in any sample we're going to have the exact same amounts of all four of these. It just means that maybe in a chimpanzee, maybe 20% of their DNA is made up of A's or adenines. That would mean that, according to Chargaff's data, the other 20% would be made up of thymine, T's. And that would equate to 40% of that chimp's DNA. But we still have 60% remaining, right? So the remaining 60% then is made up of G's and C's, and those numbers would also be equal. So I'd have 30% G and 30% C. So Chargaff created what he calls Chargaff's rules, which is that A equals T and G equals C. Based on that, the people that are going to come along here and actually identify the structure identify these rules that Chargaff created as what we now call base pairing rules. And these are consistent, as you would expect, among all living things. So wherever in a DNA molecule we have an A, we have a T that's going to pair with it. Wherever in a DNA molecule we have a G, we're going to have a C that will pair up with it. And so knowing of Chargaff's work, there were several people that come along and try to kind of say, okay, then what's the structure going to look like? Because they didn't know, what to expect in terms of how all these things kind of piece together, right? It's like a big puzzle that they're trying to kind of pull apart one part at a time. So the first person that comes along, or the next person I should say that comes along, is a lady. This is the first time we see a female in science here, which is great, named Rosalind Franklin. She was an X-ray crystallographer, which basically means you take molecules, purified molecules, isolated molecules, and you create crystals with those. And then you expose those crystals to X-rays, and those wavelengths of light that make up those X-rays refract and will bounce off of those crystals in certain patterns based on the structure of the molecule that makes them up. So it sounds super complicated, and it is. It's also not a technology that we continue to use because, as you remember before, X-rays can be super dangerous. All right, they can mutate DNA, they can change DNA, and they can cause things like cancer. Well, Rosalind Franklin at the time, this is in the 50s, the 1950s, she's using these X-ray images as part of her research all of the time. And one of her major focuses was trying to identify, what she could about the structure of this molecule that we know as DNA. And the image that she pulled from one of her samples was the one you see on the right. This is actually called photo 51. I'm guessing because it was maybe her 51st photo of something. But this is the best one that she was able to take. And to us, it probably looks, and to you guys, it probably looks a little bit like I can't tell what that is. Like, so what? But what you're looking at here is actually a DNA double helix, where we're focusing kind of on the center of the helix. So instead of the DNA double helix twisting and going vertically, we're looking at here, taking that vertical thing and kind of turning it on its side this way and looking down the double helix kind of into the screen. So if you think about that, if you've got this helix that's twisting, right, going towards the screen, you can imagine how it's kind of expanding and contracting, right, as you look towards it. And so these little marks that you see here is actually the helix kind of looking down the center of that structure. So for anybody who understood X-ray crystallography at the time, this was like major. Her ability to look at this, she was the first person that was able to actually capture an image like this of this teeny tiny molecule that helped identify this thing is actually helical. It kind of spirals in its shape, right? It's not just linear. It actually forms this helical shape. Rosalind Franklin was obviously very influential, but she also ironically ends up dying from ovarian cancer several years later. And although other scientists won the Nobel Prize for their discovery of this DNA structure, she was not awarded it because at the time she was dead. And a lot of the theories go along with, okay, she used X-rays all the time. Chances are if she died from ovarian cancer is because of X-ray exposure. And she did die like in her mid-30s, so she was not very old. But she was amazing in terms of, what she contributed to what we understand now about the structure. So these two guys here are two that actually won the Nobel Prize for their work in 1953. So in 1953, two men by the name of James Watson, this guy here, and Francis Crick here, these are the first two to identify and figure out, really, to solve this puzzle of the structure of DNA. And they used Rosalind Franklin's image here to help them do that. But, oddly enough, they use it without her permission. They worked at the same university at Cambridge in England, but they were not friends. They were actually more like folks that do not get along, not so much enemies, but they didn't want to work with one another. So they gained access to her image, and then they created the first DNA model, I should say. I was going to say molecule, but model. And they did it using lots of different materials that they built while at Cambridge. And so this is a picture of them with their lovely DNA model, and you could see how it twists and spirals kind of up. This is one of their original drawings of, how they were kind of mapping out that and where they thought, the sugars and the phosphates should be and where the nitrogen-containing bases should be. So they built the first three-dimensional model of DNA. They used the work of both Erwin Chargaff in terms of identifying those base pairs and Rosalind Franklin for the three-dimensional structure. And the two important pieces that they were able to pull from that in terms of what they actually did is they identified that sugars and phosphates go on the outside of the ladder, and the nitrogen bases kind of sit in between. So the nitrogen bases, if you think about a ladder, these are the rungs or the steps of the ladder, the things that you step on as you're moving up or down. And those are, again, comprised of these A's, T's, G's, and C's. The sugars and phosphates on the outside are going to be super strong, and that allows the molecule to stay together. All right, so in looking at this structure, this is that three-dimensional kind of image of what they would have created. So we have two strands of DNA in this double helix, and they're running in opposite directions. We actually call them anti-parallel. And the reason why they do that is they're kind of orienting their bases in a way that allows them to interact better than if they were both facing the same direction, okay? So the sugars and phosphates may alternate going down this way on one side, but then they kind of run upwards going on the other side. The base pairing rules help to identify those interactions in the middle. Again, they make up those rungs. And here my sugar phosphate backbone is just represented by what looks like this little blue ribbon. So really inside that blue space, you'd have sugars and phosphates alternating throughout. But again, A's pairing with T's and G's pairing with C's. And so that helps to kind of conserve a sequence when we look at something like DNA structure. The last thing we want to look at in terms of this structure is how those little rungs, those steps of the ladder, are actually interacting with one another. So I mentioned already, in a nucleotide, within one of them, like in this example right up here, this top one, all of those nice connected lines are all covalent bonds. So within a nucleotide, the sugar, the phosphate, and the nitrogen base are all held together with super strong covalent bonds. But what we find in DNA is that in between my base pairs, inside this molecule that makes up the rungs of this ladder, we're not seeing covalent bonds. Instead, we have some actually very weak bonds. We have hydrogen bonds. And if you remember anything about hydrogen bonds from chapter 2, these can become really important, especially in things like water, but they are very weak. They're simply based on a slight attraction, right? It's not even an ionic bond. It's not even a full kind of electric attraction. This is simply based on a slightly positive hydrogen and a slightly negative oxygen or nitrogen. And in this case, it's going to be slightly positive hydrogens interacting with slightly negative nitrogens, all right? But you can see how these things kind of form. Between G and C, we get 3 of these hydrogen bonds, which makes this interaction a little bit stronger than between A and T, where we only have 2 hydrogen bonds. And here again, A and T, 2 hydrogen bonds, G and C form 3. Now, importantly, what that means is that we can't just take an A and put it here in this place of the G. Because if you do that, they don't form bonds properly, okay? There's something here that makes A and G different, and that doesn't allow it to interact with the other molecule. So they are very consistent in terms of within a DNA molecule, G has to pair with C, A has to pair with T, and they always do so using hydrogen bonds. Again, those are kind of like temporary and partially formed bonds, I guess, if you want to think about it that way. They're very weak again. But the purpose of them is going to be to kind of hold that molecule together. But in turn, like in addition to that, We also don't want to have a super strong bond in the middle of DNA because we know that as part of the cell cycle, we need to be able to copy this molecule. So because we need to copy and access genes pretty often in a cell's lifetime, we need to be able to kind of open that DNA double helix up. And so it makes sense then to have hydrogen bonds where we can easily break them without having to exert a lot of energy and go on with copying or reading a gene or whatever it is that needs to be done. The strongest bonds are here in the backbone, and we do want to keep them there because that kind of keeps everything in place. But again, those hydrogen bonds, again, super weak, but helpful in the long run because they're good for replication or copying. They're good for reading genes, and they're also good enough to hold this thing together kind of temporarily. Okay. So that's all I've got for 13.2. Again, if you've got any questions, let me know. Otherwise, I will see you next time for 13.3. Bye, guys.